You, me, and HIFMB. Stories of Science and the Sea.
Hello everybody and welcome to the next episode of the HIFMB podcast. HIFMB stands for the Helmholtz Institute of Functional Marine Biodiversity, and I'm your host, Jan-Claas Dajka. In this episode I interviewed Alica Ohnesorge, who works on eDNA, which means environmental DNA, and that's a very new way of tracking species identities in the water, so which species have been in the water column at the time, a little bit of detective work. And she explains the process very well, and she gets into the pros and cons of environmental DNA and a little bit of the marketing, let's say, around eDNA. And how you can improve the method. She's a PhD at our institute, but she has been back and forth all over the world. She's been in the Seychelles, she's been in Barbados, China, Denmark, Kiel in northern Germany, or now she's here. So we go back and forth on her very exciting life. The takeaway is that she was always very brave in her decisions, for instance, to go to China, or with her decision to apply for the PhD. And then turns out being brave pays off sometimes. And Alica Ohnesorge is a great example of that. So with that, I give you Alica. Hello and welcome to the HIFMB podcast. And my next guest, Alica Ohnesorge, which is a great name, I must say. It's means without sorrow in German. Are you without sorrow all the time?
I try my very best.
Welcome.
Thank you.
So you're an oceanographer, but biological oceanographer.
Biological oceanographer, that was at least the study program I did my master in,
Okay. And you work on eDNA, so could you get into that a little bit, what that is?
I can.
Yes,
excellent. So eDNA is the abbreviation for environmental DNA. That means basically any genetic material that is released into the environment by whatsoever. So we do focus on marine animals, but it could be also microbes, or it could be plant material. So we do focus on plankton, especially on zooplankton, and that can be anything. It can be fish scales, it could be mucus, it could be parts of larvae, it could be eggs, it could be gametes, whatever you can imagine.
Yeah, okay. And you can basically trace whatever was there, like a detective.
That is the idea, and that is the ideal word, that we can track everything that was there. And
how long? If you sample on Tuesday at 12, how long can you back sample from that, or backtrack?
That is indeed a very good question.
Okay.
And we also with our research, try to answer that question. So especially the freshwater people, they did quite some research on that, and they found out that depending on factors like water temperature and pH and all that, it may remain for maximum 3 weeks, but that was in really Arctic waters, the really low temperature, where DNA degradation is very slow.
Okay, and in the tropics, in a flowing river, it would be washed away very quickly, or degraded very quickly.
Very likely. So I expect it in the tropics to be only a few hours.
Right. Okay. Yeah. Interesting. And so, if you can get a little bit on the ins and outs of the method, like what can you measure and what can't you measure with eDNA?
So what can we measure is, so in the end we try to have species lists. So where you can really say, okay, that was in the water. Okay. What you can't measure, or at least what you can't reliably measure, is abundances. Biomass.
So how much of each species was there? Like
how many individuals? And then
and biomass, you said you also can't measure.
It's difficult. So there has been studies who claim that they could, but so you have the PCR bias. So that's one thing, and then it depends on many factors which you can't really control. So if you just run into a reproduction area of a certain species, and then there's lots and lots of genetic material which you catch, but there was just probably one adult individual, and you may get many sequences in the end from that one individual, whereas so it's hard to really come back to really quantitative data. But or also health status or size, gender, that's all something which you can only get if you do the traditional sampling.
Right. Okay. That's exactly what I wanted to get into. I'll get into that discussion in a little bit. First of all, you mentioned PCR or PCR. You just said PCR something.
Meta-barcoding.
Meta-barcoding. Okay.
Yeah.
So I guess PCR now through COVID. Many people know PCR, but maybe you can explain it.
I hope
shortly.
so.
Yeah.
And now it becomes really technical, I guess. So PCR
Right.
is basically just a method to amplify your target. But you also do not amplify the entire genome, so all you have, but just a few for base pairs. So we target about 300 base pairs of it. So very highly variable sequence. Which you then compare to a reference database. So that is the idea behind it, and that every species, in the best case, has a different so-called barcode. So to distinguish them in the end. And the meta-barcoding is just that you do not target one species, but many different. So you have an environmental sample where you have loads of different species in there, and then you sequence everything which you target. That.
Okay, but you can, like you said, each species has a barcode. You can directly distinguish those species from each other. So there's not a case where it's like, oh, it could be this species or it could be this species.
That would be the ideal world if you could do that.
Yeah.
No, it's really more difficult. So first of all, you need to have a reference database.
Yeah. Okay.
So a reference sequence. So before that, you needed someone to really identify that species by morphological characteristics. And then you sequence only that species,
right.
So you have a reference, which is also really a validated one. And we also see that it also, again, very technical, depends on the marker. Some markers do quite well in resolving down to species.
A marker is the...
Markers is the region you target.
Right, okay. But not the region on the
region,
animal, but
region on the genome.
On the genome, okay, right. Yeah. So the whole process. Maybe I can sum this up correctly. So you pretty much go into the sea. You sample. How do you sample? You just sample a bit of water, or
what we do is So we sample water. So we target for depending on the project a little bit 1 to 2 liters, sometimes even more, and then we filter it.
Okay.
So we just put it on a very fine port filter to collect the genetic material and get rid of the water.
Is there any genetic material that is smaller than others, so you need to vary your pore sizes? Or?
So there might be so-called free DNA, which is then already much more degraded.
Right. Okay.
There is not yet been a standard in which pore size to take.
Okay.
We are also on that.
Right. Okay. You're right in the midst of it.
Yes.
Yeah.
Absolutely.
And then so you filtered the water, and then?
And then we do DNA extraction. So we do want to get rid of the remaining salts and all the
from the filter.
From the filter that was in the seawater, and all the cell material we do not need, so proteins and and all that stuff.
How do you do that?
There is commercially available DNA extraction kits.
Okay.
So what you do is there's a buffer in there, and also basically a lysis buffer.
Okay. And
this opens up the cells so that the DNA gets free. And then there's usually you have spin columns, so silica-based spin columns, where the DNA gets stuck on.
Okay.
And then you have an elution buffer which you can pour on, and that one again elutes the DNA. Then
elution.
Elution.
With an E.
Yeah.
Yeah. Okay. All right. And then you from that you can then.
Then you have your template material, and from that you can do the PCRs.
And so. That sounds like a lot of lab work. Do you do that yourself?
We did it ourselves, yeah.
Yeah. Jeez, do you like lab work? Is that something for you, or?
I liked it very much, but I must also admit I also liked then the morphological analysis. So really having a look on the species themselves. So it was really cool to having both methods. And
but you didn't catch the
no,
species?
no, not in eDNA. What we also did for some of our projects that we took also net samples. So we towed a zooplankton net.
Oh, I see.
So we caught the animals to also have a kind of like backup proof to really see so what we do have in the nets and then what we do have or what we get with the environmental DNA metabarcoding, if that is more or less the same.
Right, okay. And so you're doing a PhD right now at HIFMB?
Correct.
Yeah. And you're writing up at the writing up stage?
Yeah, I'm still about analyzing and writing, started writing.
Yeah, you joined in 2019?
2019, yeah.
Yeah, okay, right. And so what's the general project about, or what's the aim of your project?
So my general aim is to understand actually environmental DNA metabarcoding and also improving the method because there's many uncertainties.
Yeah, as you mentioned, I've heard a bunch now.
yeah, and there's probably even more.
All right.
So that's basically it. So and then the overall aim, but that's not possible within one PhD study, is to really standardize the method and to make it available for whoever wants to use it.
Yeah, right. Okay. So we've said in the beginning that we're going to get into that discussion. In my work, I work a lot with stakeholders, and I kind of get the vibe that some of them think that eDNA is going to replace standard or classical sampling in the future. And maybe you can speak a little bit. What's your opinion on that? Because I think if you can't measure abundances, then obviously it can't ever replace classical sampling, maybe. Or...
Yeah. I'm actually on the same opinion with you. That's an absolutely problem, and also how to compare the eDNA results we got now to the results. So especially if you do if you have long-term data available and you want to compare that, how would you do that?
Yeah, exactly.
And just by coming up with species lists, which you can't even be sure if that species was really there or the genetic material of that species was just transported from anywhere else to that area.
Right.
So you can't even really be sure that species was really present there.
Yeah.
At that
time. So the results you get at this point are literally species lists. So lists of different species names that were there.
Yeah.
Okay.
Yeah.
Okay. Yeah. Do you think maybe eDNA can improve to getting abundances in the future, or is that something that you don't think will ever happen?
Wow, that's a very good question, and it's also highly debated right now.
Okay.
So I think it will improve at least a little bit, and so my PhD sister, Sarah Taudin, is a little bit working on that.
PhD sister, I like that.
Siblings.
Yeah.
So she will do a PCR-free method. So she will do shotgun analysis. So the problem is why we can't have the abundance data is... So one thing is that we do PCR, and PCR always has a bias.
Yeah.
So it can be that now it becomes very technical again. Sorry for that.
So okay.
the primers we use. So the in the end the enzyme that amplifies your target sequence needs to have kind of like anchors. So it needs to know where it has to start and where it has to stop.
Yeah. Okay.
And for that you need so-called primers. And these primers do not fit equally well to all species.
Okay.
So it may be that it favors a
so they fit some species better than others, right?
And then you get more reads, so more sequences for certain species, while others are underrepresented.
And that's a PCR bias.
That's a PCR bias, and we have huge problems with that. And you do avoid that by doing shotgun. And
what is shotgun? It sounds wild.
It sounds wild. Yeah. What you do is you do fragmentation of the genome, and then you also do sequencing. But you sequence more or less everything you have in there without knowing what is in there.
So like a shotgun shot. So
exactly.
You just spray it randomly. Yeah. Cool. Interesting. So maybe we can, or maybe I'll ask Sarah when she's on the podcast.
Yeah, she's the expert.
More on this. Yeah, excellent. So, and where were your field studies mostly during your PhD?
In the North and Baltic Sea.
North and Baltic Sea? Yeah. Do you like one better than the other?
It's really hard to say. Because I did my studies at Geomar, I'm more the Baltic Sea fan.
Yeah, me too.
Yeah.
Yeah, I'm from the east, so I
Okay.
need to like the Baltic Sea.
Yeah, but North Sea, so the North Sea does have more species, so it's hard to compare. It
does? Okay, I had no idea.
Yeah, especially so the further north you go in the Baltic Sea, the less saline it gets, so the salinity decreases. And there is many species which can deal with that, so the diversity decreases.
Yeah, okay. All right. And so part of your study was in Helgoland, I think I read?
Mm-hmm.
Yes, okay.
Yeah. That's where it started. So our focus group just had to establish and is now growing and growing. And at that time it was only Silke Lagman, Zara and me. Mm-hmm. And we have been to Helgoland in 2019, also in summer, and did our sampling there.
All right. So when you sample an eDNA sample and you get a mass of the same species, like you get a lot of readings of the same species, are there like equations to extrapolate to biomass, or is there anything where you can get from your readings to assuming how much biomass there was?
So there were studies who tried that, but they didn't do it in the real environment. So they did mesocosm experiments. Mm-hmm. And there they also had a very, very limited amount of species. So usually fish species and only 2 or 3 species within one tank. Yeah, right. And also species that behave very equally. So either only demersal species or only really pelagic free swimming species.
Okay, so it's quite limited.
Yes.
Yeah. Okay. And you just mentioned the eDNA team of HIFMB. So it's you, Sarah Taudien. Mm-hmm. Yeah. Okay. And Silke Lagman.
Yeah.
And
we grew bigger.
Yeah.
just very recently. No, we have Nora Bending.
Oh, Bending. Okay.
Mm-hmm.
All right.
But you might have not met her so far because she's
exactly. So
busy in the lab in Bremerhaven at AVI.
Okay, so she's the lab technician? No.
She's our lab. Yeah.
Ah, right. Okay. And I guess.
And then very recently we got 2 new postdoc positions. Mm-hmm. So Bang.
Yeah.
And Kerstin.
Okay. What are their last names?
Kingsley Chu Bang.
Oh yeah. Okay.
And Kerstin Clem.
Okay. All right. Cool, and they will be taking on your legacy of improving eDNA or getting more into the ins and outs, or
so they are also in other projects. So focusing on one project is more about the European oyster species and its redistribution in the North Sea.
All right,
and the other project is also more to really get it. For stakeholders and to really establish the method a little bit more. Yeah,
excellent. Okay, I was wondering with that stakeholder thing, that stakeholders have such a maybe a wrong view of eDNA. Maybe do you think that's because of bad marketing by eDNA, or?
I think our marketing is even too good, actually. So that it kind of pretends being something that can just very easily applied and everything is fine.
Yeah, yes.
So I think we still live in our pink bubble. And
but I mean, it is a super helpful method and a super modern method.
Absolutely, for sure.
Yeah,
we were so even we were really positively surprised that we get that many species. So just from the Helgoland project, we had more than 200 species by environmental DNA, which is really quite a lot.
Yeah, I mean, it's a very efficient way of sampling species richness, for instance.
Absolutely.
Or something. Yeah, exactly. So and I guess it is hard to downplay that it is so useful. So then marketing becomes tricky.
No.
But absolutely, I think it's a phenomenal technique and definitely here to stay for sure.
Yeah, so it really has a lot of potential. It's really cool, and also it's super easy, just sampling a few liters of water and getting a species list in the end.
Yeah. Do you want to stay in it? Do you want to keep working on eDNA, or?
Oh, very good question. In general, I can say yeah. So I'm
a bit hesitant.
A little bit. No, I do like working with in general the biodiversity assessment, and that's quite a good method to doing that.
Yeah, absolutely. Like we just said. Okay, and maybe let's dive into your life a little bit. When did you first come in contact with eDNA?
So with eDNA, actually, really here. That was really so. I have had heard of it already in my studies, in my master's studies, but didn't get really into contact with it because it was so super new and no one working with it at the institute. I was, at least not that I was aware of it.
Mm-hmm. So what or maybe let's go through your life chronologically, through your academic life. Yeah. Bachelor's. Yeah, where did you start?
I did my bachelor degree in Göttingen at Göttingen University at the Georg August University.
Are you from Göttingen?
I was born there, but grew up in a very tiny village.
What's
kind
it
of
called?
Pulde.
Pulde?
Yeah.
Okay.
Well, so a little bit nicer. So Herzberg am Harz is kind of like next to it.
Yeah,
and
Harz is a really nice region in Germany.
Yeah, I was more referring to the name because Herzberg, and then my last name, and then it's always when you did. Yeah.
Okay, right, I get it. So okay. But yeah, Harz, I love it for everyone or anyone who doesn't know, Harz is in Germany. So we're talking about
in the kind of like middle part of Germany.
Yeah, it's a
mountain area.
Yeah, mountain range in Germany. It's super nice for hiking. Have to go.
Doesn't have a lot to do with ocean or sea.
No, it doesn't. Yeah. When did you catch the bug, the ocean and sea bug?
During my bachelor degree, because I did my bachelor thesis. I was so super lucky that I had the chance to go into the Seychelles.
Oh yeah.
Yeah.
Excellent.
Yeah.
When?
2013.
Okay.
Already quite some time ago.
Yeah, I've been
10 years almost.
I've been in 2017, 2018.
Ah, cool.
Yeah.
Nice.
Where did you go?
To the main island.
Ah, okay, Mahé. Yeah. And doing what?
Working on corals. So we have been, so we went to the coral reef there and then documented what species live there and
I had no idea. I did the same pretty much.
Wow, cool.
Just for a PhD project, but same story. Cool. And did you like Seychelles? Did you like Mahé?
Yeah, it was so nice. That's really like paradise on earth, right?
Yeah, it is. Oh man. Yeah. Did you see any of the Aldabra turtles, the big giant turtles? I think it's the largest land turtle on Earth. Yeah.
I saw them, but not in nature, just in a.
Oh, okay.
Well,
yeah.
At least I did.
Yeah. There's few left.
Yeah.
Okay. And then bachelor thesis in Seychelles, and then back to and then you wanted to go to the sea, work on the sea.
During my bachelor's, I was already quite sure that I would continue with something like zoology, ecology, like something like that.
Yeah.
And then having...
Sorry, I'm not sure I asked. What was the degree called?
Just general biology.
General biology. Okay.
Just very...
All right.
very general.
Yeah, okay.
And that kind of like caught my attention to that fascinating ecosystem, the ocean. Yeah.
Yeah,
and that's how I decided to continue further.
And where'd you go?
After that, I went to the Ocean University of China in Qingdao.
Oh.
Yeah.
Göttingen, Seychelles, China.
Somewhere very far away.
Yeah. Do you speak Chinese or?
I wish I could.
Okay.
No, very, very. So I do understand very, very, very simple words, and not even sure if I still do.
Yeah. Right.
Yeah.
But that was sort of a double degree, I think.
Yeah, so the University of Bremen and also the University Kiel, so GEMA, has that you go there for 1 year and then you study marine science. And afterwards you return back to Germany and do a regular studies at your university home institution.
A year in China, how was that?
Super difficult and super different. That was so my main problem, guess, was really the language issue.
Yeah,
because I was a little bit too naive, maybe going there and.
It's brave. I think it's pretty brave.
I was just thinking, okay, maybe so. Well, so I do not expect everyone speaking English, but I expected everyone speaking English at the university, especially as I did an international degree there. So the the degree was also in English.
Yeah, okay.
But it was really tough. So even with those lecturers we sometimes really had difficulties to understanding each other.
Oh, okay. Even in English?
Yeah.
Okay. Oh, good.
Yeah.
Damn. Okay.
But apart from that, it was quite a cool experience. So I benefited a lot from it, I guess, because it's so different and it's just...
Yeah, in what way did you benefit from it, you think?
In kind of like flexibility and to, also handling different cultures, very different
openness.
ways of thinking. Yeah.
Cool.
Mm.
Well, you're definitely open to
Thanks.
to podcasting.
Yeah, for that it is.
Yeah, no, serves me well.
Hope so.
Yeah, and so then 1 year in China, and then you came back for 1 year in Kiel, was it?
It was, so the normal thing, I did 2.5 years in Kiel. Mm.
Yeah,
at GeoMa.
Yeah, in the north of Germany, Kiel.
Yeah.
And in that you said, or you didn't say, I read in your CV that you also did your master's thesis in Denmark then? So it's like the triangle of China, Kiel, and Denmark.
Kind of, yeah.
Yeah, okay, right. Cool. And what did you do your thesis on?
My master thesis was so I did work on an invasive ctenophore, so common jellyfish species that had been introduced quite some years ago to already the Black Sea and also Caspian Sea, and now also found in huge amounts in the North Sea and Baltic Sea. And they are really invasive because they're like tiny feeding machines, really feeding on everything they get, and they can be quite disturbing for the ecosystem. And I did my research on if they could also adapt to the low salinity regime in the Baltic Sea.
And can they?
They can.
They can.
So maybe not the lowest, but
okay.
So the Black Sea and Caspian Seas have much lower salinities already. So they, depending on where you are, even as low as 5. So that's already very low. And in the Baltic Sea, so in the German parts, it's still higher. So at Kiel, it's like 17-ish or even higher, depending on how much water from the North Sea gets in. Yeah,
I just know from the North Sea where I work, it's around like 23, 25. A PSU, I think, is a unit.
Yeah, I think there's also debate. I think the chemists even do not have units anymore because there were some... Yeah, but PSU is still used very often, so I think it's okay.
As you can tell, I'm definitely not a chemist.
So and the species thrives actually in at this lower salinity areas in the Black Sea and Baltic Seas, and so we wanted to know if this other subpopulation could also do the same in the Baltic Sea. So we had 2 different generations where we treated them in different salinity areas, and they already improved during that time.
And when was this?
Well, in 17, 18.
2018, okay. And do you know what the situation is since? Or
actually, I lost a little bit the contact. So what I can tell you is that at least the species didn't make it to establish in the very far north areas. So we are not there yet.
Okay.
But you can frequently find them also where we did now our sampling. So I had another project where we sampled also at the Lim fjords in Denmark, and also in the Western Baltic Sea in the German parts.
All right.
And I had loads amount of them. So in our eDNA reads as well as also in our zooplankton net. So they are really abundant there.
All right. Now you're keeping track of them via eDNA.
Yeah. They can't get rid of me.
They can't. No.
I will chase them forever.
Interesting. And so I read in your CV that you're quite the cruiser.
The cruiser.
You've done many scientific cruises.
Yeah.
Do you like it?
I love it.
Yeah?
Yeah,
Oh, really? Okay.
I really wish I could have done more, but COVID situation.
Oh, man, I think you've done... Man, sorry.
Yeah, no, I still got lucky, and I started. So my first cruise was also at Jeyumar, actually, where I was on a fisheries cruise.
Yeah, okay.
Where I had to, cut fish and, well, let's talk about something else.
Did you not like it? Cutting fish?
I was not really.
Understandable, I think.
It's a little bit okay. So, I mean, it's all for science, and we all need these data, but just seeing, like, the fish and which you come on board and which are just, going back into the sea afterwards without any need, so we do need it, but I didn't like to see it.
Yeah, okay. But the rest around it, like the rest of the cruise, you enjoyed?
Absolutely, for sure.
Okay. How long was it?
That one was for almost 3 weeks in the Baltic Sea.
Right. Okay. Mm-hmm. Through the whole Baltic Sea?
Mainly the German parts, because that was funded by German sense. And to just see how the fish stocks do in German waters.
Mm-hmm. And then what was your longest cruise in your life? I mean,
actually that one.
That one? Okay, all right. Yeah, I've only been on one, and I think it was a month, and that's definitely maximum time for me. I need more space to run around, I think. It becomes kind of like you have to be the flexible type.
The flexible type.
Which you definitely are, I think, based on what I've heard now. And then you've also been on you've been doing your double degree. You've been in and out of various projects, and one of them was in Barbados, I think.
Yeah, I really chose the nice places, right?
Yeah, you did. Hey, for sure. Yeah, how was that?
It's super nice. No, I really got lucky because at my time at Jeyumar, we were also supposed to do another project somewhere else. Mm-hmm. And at that time, I was like, actually it would be cool to because due to also private things, I searched for an institute doing marine research in Switzerland. Mm-hmm. And Switzerland is not the place to be for...
No, it's absolutely landlocked.
But I still found a working group doing research on fish behavior.
You did?
I did.
Very resourceful. Yeah. you find them.
Yeah. No, that was really lucky coincidence. Because at that time there was a PhD student, Renata, and she looked for someone who could help her for her PhD project on Barbados.
Excellent.
And having a diving license, and I was here. Am I?
Oh, you do? You can dive?
I do not have the scientific diving license. But,
Right. Yeah, you've obviously then been diving on the Seychelles as well.
Yeah, we did most of it by snorkeling, actually. But we also did some nice private diving.
Yeah, right. And then, so you did have your divers, not drivers, divers license.
Divers.
Yes. And worked on gobies, I think.
Yeah, and cleaning gobies.
Cleaning gobies. They're so cute. That's ridiculous.
Yeah, I love them so much. Yeah, maybe a little bit. So usually, cleaning gobies, so there's different taxa. So most of the cleaning fish are wrestles, but there's also cleaning gobies. And these tiny guys. So she did her project on a certain species, and that species has 2 different ecotypes.
What's it called, the species?
Electinus proctylus.
Oh, right, okay. Mm-hmm. I thought it would have an English name.
Maybe it does, but I do not know.
Okay, all right.
Might have been.
Yeah.
And
but remembered. I don't remember any Latin names of anything I'm working with.
Yeah, and that species has 2 different ecotypes. So there's either... they live in pairs, so female, male, and so Renata called them coral dwellers. So they really live at corals and then waiting for a fish to pass by and then feeding on the parasites or also stealing some mucus or scales from that fish. And the other ecotype is that the very same species living in bigger groups in sponges, mainly basket sponges, and then just feeding on any invertebrate they might get.
Oh, within the sponges?
Within the sponges, yeah.
Right. So they don't have that cleaning behavior? Or?
No.
Okay.
For any reason. So they might switch from time to time, but usually they really stick to that ecotype.
That's interesting. interesting.
Yeah, it was. I also wasn't aware of that before.
Yeah.
And she had the hypothesis that these pairs, so the ones that actually clean, would do better in recognizing patterns, just because to really recognize those fish they can clean on.
Yeah.
And the sponge-dwelling ones would be better in recognizing, so they would be better in spatial tests, so to remember where they have to go. In the end, we didn't find any difference, but only that also, no matter which ecotype they were, they did both better in the spatial tests.
Sick. Okay.
But they were really, it was really cool because we really trained them to either a certain pattern or a certain, like either left or right.
Right.
And it was really nice seeing them really improving in there. And they were really like, okay, I want to learn, and I want to have that kind of,
Oh, that's awesome. So with with all these little side projects going on during your masters, did you purposely do that to like find a place to specialize in or topic to specialize in, or did you want to end up in eDNA and oceanography, or?
I didn't specifically wanted to end up in eDNA. I think I was just curious to look in different projects, and also when I did a research project somewhere else, so especially one also in Australia, I also had a look on what other PhDs are doing and having a look on.
Oh, in Australia, I think we haven't talked about this.
Not yet, no.
No, we haven't.
The Australian, that was also with a PhD student and Becky, so Rebecca, and she did her project on, so she looked on rock pools that and actually, so what species live in rock pools, and then because there's this also due to the engineering and also because building sea walls to protect beaches, the rock pools kind of like disappear. So they like really natural occurring ones,
right?
And she wanted to replace them kind of like with flower pots. So she just installed, it was really nice. So she just installed flower pots on seawalls. And then I had a look which species settle there and if there was a difference between the naturally occurring rock pools and the flower pots.
And?
There was, but so it really took some time because you also have like sessile species like bryozoans and that just need some time to settle in these flower pots.
Yeah, okay.
But during her time of installing them, it really became more and more diverse. So it was really nice to see. And she was always like, oh, I really want this sea hares, these bigger snails.
Big underwater slugs, I guess.
Yeah, they're slugs, basically. Yeah. I really want to have them in my flower pots.
Oh, that was.
I guess she never had, at least not at the time I was there.
Oh, she did. Okay. How long were you there?
For a month.
Okay, interesting. Did you? All right, and okay, coming back to the original question. So you didn't want to specialize in the topic. It was just about the curiosity and getting experiences in a broad range.
Yeah, I looked for projects, working with marine science in general and also with kind of like zoology, ecology, and also this behavior project because I've never worked in behavior biology before, and just to just getting a little bit an idea on that.
Mm-hmm. Awesome. And then you saw the application process for this PhD and just went for it, or
I was just also a little bit brave and went for it because
absolutely.
Also a little bit like the naive part of me, maybe as same as going for China. I was like, okay, I do not have that much experience with sequencing. Let's just keep trying. Maybe they do want me because sure. And they did.
Yeah, exactly. Obviously, you're doing well with it. So congrats for being brave.
Thank you. Was worth
it. Yeah, definitely. Oh, it's. I think it's worth it. I wouldn't call it naive. Yeah. All right. So then now you're writing up your PhD thesis and hopefully that goes well.
So far it does.
Yeah? Okay, perfect. Then I wish you all the best with it.
Thank you so much.
Yeah. And anything, so we're at the 40-minute mark already. Is there anything that you think we've missed or that you want to raise or that you want to mention?
Just for, because we just talked about it now for, I think, 2 times or 3 times. Maybe just so stay curious and also look into different topics which are may seem far or further away, at least from your own topic.
Yeah, that's just perfect advice. Yeah, I love that. Thank you very much for coming, Alisa.
Yeah, thanks for inviting me. That's really good.
Awesome. And see you in the next one. Bye-bye.
Bye.
Want to dive deeper? Surf over to hifmb.de or follow us on Twitter at hifmb_OL.